lecture. Okay, we are recording now. All right, a couple of announcements that I have to make. You will have two opportunities to actually take the final exam. One of them is Saturday, December 14th, 1:30 to 3:30 in this room. Okay, I had some emails about exam 3 as well. How can we retake it? Here's what we'll do. It's a 2-hour block. Exam 4, which is going to include acid-base regulation, GI physiology, and endocrinology, is the first hour. The format is exactly the same. You'll have an hour to do that exam. The second hour is devoted to any retake that you want to do. Usually we have exam 1 on this side of the room. Exam 1 will retake that exam. The second hour of the final. The exam 2 will be in the middle, and exam 3 is usually on the far side there. Yes? Taking them with a group, yes. So that is a group exam, by the way, all right? So what is going to happen is you all can talk about the makeup exam. You can work in a group. The tables can work together. The only thing you can't do, right, is when you take a look at the exam over again is you can't memorize A, B, C, or D. Only because the questions will be a little bit different, okay? So you really do have to think about that exam, restudy it, and then, again, it's not going to be exactly the same. It's going to be very similar, but it will be a group exam, yes. Okay? Actually, the vet school does that now. The vet school allows you to retake an exam, and you actually get to work in a group. All right, any questions about the final? Yes?
Well, I was just wondering, like, if you're taking a paper exam.
Yes, you can come into my office and take a look at, you can take a look at your exam, Usually I have like a conference room right before the exam that you can come in and take a look at it again. Yep. All right, the second opportunity to take the exam is going to be Tuesday, December 17th, 1:30 to 3:30. I couldn't get this room, but it's going to be in, hold on just a second. Let me make sure I know what this is. Let's see here. I'll have to tell you, I think it's in actually 205 Hacker Hall. 205 Hacker. But I did reserve that room. I just can't remember which one it is now. So I'll make that announcement. I'll just make an announcement on your Canvas website later today. All right, pretty good? Yes, question? Yeah, I'm going to actually open that up so you can see it again. The other thing that I would really, I'm actually, still debating, but I'm going to try to make this a rule, is the final exam and the makeup is going to have to be on proctorial. Okay? If you have some issues with that, like your computer really is going to crash and you just can't, you just can't take the exam on proctorial, then please come and see me. We can make special arrangements. But it's just too much with the final exam and the retake exams. That grading is going to be just like really intense for Meghna and I. So, okay, any questions, any concerns? Okay, so again, you'll have two opportunities to take the final exam and the retake. That's going to be Saturday, December 14th, 1:30 to 3:30. Or December 17th, 1:30 to 3:30, and I'll announce that room later today. I think it's 205 Hacker Hall. All right, pretty good? Okay, again, we're going to start with GI physiology. I was going to say one more thing. Oh, yes. I did have some questions about retaking the final. And that's the only exam that you can't retake because we probably won't be grading that until after the 17th. So there's just no way to retake the final, okay? All right, so let's go ahead and get started here. I really like this particular section. In the past, the previous professor before I started, they didn't even teach GI physiology, which I was shocked about. We have a lot of nutrition students in this class, so we want to make sure that you all have this interesting information. The GI tract is actually an organ system. It's made up of a number of different organs that all have the same function to digest and absorb nutrients, okay? So digestion of food and absorption of nutrients are accomplished in a long tube connected to the external world at both ends, okay? Secretion and absorption happens in the gut, and these are major themes in understanding the small intestines, large intestines, this tube, okay, the tube. This is also called the alimentary canal. Alimentary canal, it's A-L-I-M-E-N-T-A-R-Y, alimentary canal. The idea here is basically if I stuck a tube down my mouth and I wound it through the digestive system and then out my anus, butt, right? Nice talk here on a Monday morning. That is all considered the alimentary canal, okay? So take a look at this. There are, again, just like we talked about with the kidney, 4 different processes carried out by the GI tract. We have digestion. Digestion can include chemical digestion, mechanical digestion. Your stomach does both. I'm going to talk about this later on, but the stomach is actually a pretty violent place. It is very low in acidity after you eat a meal. It can be a pH of as low as 2. And also you have these different types of muscles. In fact, 3 different types of muscles that churn the stomach, circular, longitudinal, and obliques, that help to mechanically digest food. Okay, so digestion, secretion. Now again, secretion is just like what we talked about in the kidney. Anytime you move solutes or fluid from the blood supply, the circulatory system, into the intestines, that's called secretion. Absorption goes in the opposite direction, just like reabsorption in the kidney. Absorption pulls nutrients and fluids out of the gut and into the circulatory system. Okay? And then motility is actually another major theme, and we're going to be talking a lot about motility, whether we're talking about peristalsis, segmentation. So there's many different types of motility in the gut to try to keep moving that food down the alimentary canal and then undigested food out your body. All right, so let's take a look at some of the major organs that play a role in the digestive tract, the GI tract. There's many functions in the gut found in very specific locations along its length. Most of the absorption of nutrients occurs in the small intestines, so most of the digestion is accomplished there or upstream. So we'll talk today mainly about the upper GI tract, which is up here. I did go to a national meeting a few years ago, EB, experimental biology, if you all have heard of that. And I went into a lecture. It was a refresher lecture on GI physiology, and the gentleman was just ranting about how physiologists don't give enough time to the upper GI tract. So I am actually going to take a little time to talk about the upper GI tract today. Then we're going to get into the esophagus. Mainly the esophagus just moves food from the mouth region into the stomach from A to B. But then in the stomach, it's much more complicated. We'll talk about parietal cells that are acid secreting cells, chemical digestion, motility, as I mentioned before, and then we'll talk about the small intestines. We'll get into the different segments, duodenum, jejunum, ileum, and most, again, of the absorption occurs in the small intestines here. So now you've probably heard of some of these other organs. Some people are not quite sure where they are or what they do. So we're going to take some time to talk about the pancreas as well. It's a very important organ that helps with digestion. It delivers pancreatic enzymes that help break down carbohydrates, lipases, proteins. It delivers proteinases, which we'll talk about. It also delivers bicarbonate. Okay, so this is one way that you can neutralize the acid that's coming from the stomach. So there's a huge dump, basically, of bicarbonate after a little bit of food enters into the small intestines to neutralize that acid coming from the stomach. Also, the gallbladder. Actually, that's a very important organ as well. If you, there's some people in here that are in the lab. If you lift up the liver when you're opening up the fetal pig, you'll notice a little green kind of pouch that's right underneath it. That is the gallbladder. And the gallbladder delivers bile in the same, actually across the same duct that the pancreas delivers some of their enzymes, the sphincter of Oddi. And basically what it's delivering, bile, is it's an amphipathic molecule that helps to emulsify fat. It helps to emulsify fat so lipase can get in there and break fat down. All right, so we'll learn all about that as well. Then we'll get into the large intestines, the colon. You can actually see that the colon looks, morphologically, it looks much different than the small intestines here. We'll talk a little bit about the appendix, cecum, ascending, transverse, sigmoidal colon, and then finally the rectum and anus. All right, we'll talk about the defecation reflex. As you probably noticed, the gut is not very glamorous. We're going to be talking about vomiting and diarrhea and all that good stuff. So it's fun for me, not always fun for you this early in the morning. But yes, we're going to get into all of that as well. All right, so I do want to talk a little bit about absorption and secretion of water. Digestive secretions are mostly water. And I want you to notice that only about 100 mils of water is actually lost in feces. So the mechanisms for water absorption are efficient, and you have to remember the kidney and its role in ion and water regulation. Okay? All right, so take a look at this. You usually, on average, ingest about 1.2 liters of water per day, or you should anyway. And that includes some of the solids that you ingest also that have a lot of water in them. You'll notice that you also secrete about 1.5 liters of salivary secretions on a daily basis, and about 2 liters of gastric secretions that occur in the stomach. Whenever you hear gastric, you can just automatically think that that pertains to the stomach. The gallbladder and pancreatic secretions together actually are another 2 liters. So, you know, that is 2, 4, let's see, 5.5, about 6.7 liters, right? Of all of this are secretions. Now, your small intestines, as I mentioned before, they absorb about 6.7 liters. And they deliver that back into your blood. Okay? Then you have about 1.5 liters of intestinal secretions, and then again, your colon absorbs about 1.4 liters. So again, you only lose about 100 mils of water with your feces. All right? So I want you to also think about cholera, some of these pathophysiological diseases. Cholera, in particular, actually, instead of absorption in the small intestines, what you get is secretion. All right? So imagine now with cholera, you can't absorb almost 7 liters. It's actually starting to secrete. It's no wonder that with cholera, you can die within 24 hours because you lose almost all of your bodily fluids, right? That's why it's so devastating. Instead of absorption in the small intestines, you get secretion. And you can lose your entire bodily fluids within a 24-hour period. All right? So just something to think about. We'll talk more about cholera a little bit later. I'm going to bring back CFTR. Remember cystic fibrosis. Cystic fibrosis actually this is a hypothesis, may have persisted throughout history only because it gave some protection to people that actually had contracted cholera. Okay, so think about that. It's interesting. We'll talk more about that a little bit later. All right, so more about specialized compartments. I have actually talked about all of the different organs, the major organs that we're actually going to be going over. But let's talk about why they work individually and how they work as a common function of digestion, absorption, and secretion. These specialized compartments are actually divided by these muscular valves called sphincters. These sphincters control the passage of food from one compartment to the next. This allows each of these compartments, the esophagus, the small intestines, the stomach, it allows each of these compartments to actually vary in pH, enzyme composition, and types of cells, whether they're secretive or absorptive. Okay? So that's how the digestive system works. They have these different compartments that do different functions, and they're divided, they're controlled by these sphincters, these muscular valves, okay? So there's a lot of complexity within the gut, especially across different species. And again, we'll get into some comparative physiology also in this section. All right, so here's kind of how it looks all together. We actually are going to be talking about the sublingual, submandibular, and the parotid glands this morning. So again, we're going to start by looking at the upper GI tract and all of the salivary glands. Notice again that here's the mouth region. The back of the throat is called the pharynx, and you have that epiglottis. We talked about this before when we talked about the respiratory tract. That epiglottis actually protects the trachea when you swallow food. That food should enter into the esophagus and then travel to the stomach. Okay, so kind of giving you some orientation in some of the different structures that we're going to be going through. All right, so before we do that, I do want to go and talk a little bit about the teeth, believe it or not, okay? The teeth is a big part of what's called mastication. Okay, so I do know my animal science students know about mastication. Mastication is actually just a really fancy name for chewing, okay? Chewing food up. This is already a type of mechanical digestion, kind of making these larger bolus of food into these smaller bits. You actually have 12 molars, 8 premolars, 4 canines, and 8 incisors, okay? And that includes both the lower and upper jaw. So the incisors and the canines are about tearing and biting, where the molars and premolars are about grinding and chewing. Okay, so that's some of the function of these different types of teeth. Makes sense. Canines and incisors, biting and tearing. Molars and premolars, grinding and chewing. So I have had students in our department as well that have gone on to dental school. So we want to talk a little bit about the teeth in more detail here. The enamel is the hardest substance in the human body, and it actually contains the highest percentage of minerals. So you can see here the enamel is on the outer part here. 96% of enamel is minerals. Only about 4% is water and organic substances, organic material. Now this is a type, the primary mineral is a type of crystalline calcium phosphate. Okay. All right, the next thing is the dentin. The dentin is calcified tissue of the body, and along with enamel, cementum, pulp, it's actually one of the 4 major components of the teeth. Okay. And then we're going to get to the pulp cavity. The pulp is the part in the center of the tooth that's made up of connective tissue and cells called odontoblasts. Okay, odontoblasts, these are cells that give rise, they're stem cell-like, that give rise to new cells. And this dental pulp is soft tissue in the center of the tooth that contains, you can see here, nerves. Where's my little arrow? Here it is. Contains nerves, circulatory system, and the lymph system. Lymph vessels. And connective tissue. So the tooth's nerve lies within the root canals. You can actually see these root canals, which lie within the root, or what's also known as the leg of the tooth. And the root canal travels from the tip of the tooth's root into the pulp cavity. Okay, again, it also contains blood vessels, connective tissue, which nourishes the tooth. Then you have this periodontal membrane. It's also called a periodontal ligament. It's a fleshy tissue between the tooth and the tooth socket that holds the tooth in place and attaches it to adjacent teeth. And that helps to resist the stress of chewing. Okay. All right, then you have the bone here too. So the tooth can be divided basically into the crown and the root section, and the gingiva, this is the gum line here. All right, so just different parts of the teeth. Very important, you can see this molar here and grinding and chewing. All right, so let's go into the saliva. The mouth actually is the first compartment that starts to process the food for swallowing. And there are important salivary glands that I want you to be aware of. The parotid, the submandibular, and the sublingual gland. So let's take a look at that. I always do this, some students can't find it right away, but if you take your tongue and you kind of run it along your upper jawline right here, you can sometimes feel a little bulb, right? A little bulb area. That's the opening of the parotid gland, okay? So the parotid gland is the largest gland, but at rest, if you're not actually eating any food, the parotid gland contributes to only about 25% of salivary secretions at rest. All right, at rest, the submandibular gland actually accounts for almost 60 to 70% of salivary secretions. And the sublingual gland here only accounts for about 3 to 5%. And we'll be going through each one of these. All right, so let's start with the parotid gland, the first one. The parotid gland, again, accounts for about 25% of the salivary secretions at rest, but when you eat a meal, it bumps up to about 50%. All right, so what's in these secretions? Basically, the composition that's coming from specifically the parotid gland is mucins, amylase. Amylase is something very important that I want you to remember. Amylase is an enzyme that already starts to break down carbohydrates in your mouth. It's called salivary amylase. Also bicarbonate to neutralize acid, and lysozyme. Lysozyme is an antimicrobial. So you're already kind of knocking down some of those microbes that you've eaten with your food. All right, so when you eat a meal, the parotid gland ramps up to about 50%. Submandibular gland ramps down to about 47% or so. The sublingual gland is mainly mucus secretion, mucins. Only about 3 to 5% of the salivary secretions come from the sublingual gland. One thing that I always remember the sublingual gland, if you think about, my dog is a huge yellow lab, but I think about St. Bernards too. Think about that mucins that have you ever seen them when they're hungry and they're looking at your food and all of that secretion, just my dog in particular, he can secrete mucins that almost touch the floor. It's pretty amazing. I'm going to have to take a picture of it. But that's all, I'm sure, coming from the sublingual gland, right? All those mucin secretions, all right, which is ramped up again when you eat. So these are multicellular exocrine glands. We're going to talk more about exocrine and endocrine glands later on, but an exocrine gland basically delivers its substance into an open cavity, right? So these are exocrine glands that deliver salivary solution, salivary fluid, into an open cavity like the mouth. In contrast, an endocrine gland delivers its hormones or molecules into the circulatory system. Okay, so these are multicellular exocrine glands. The saliva actually helps to lubricate food, digest carbohydrates. Polysaccharides are carbohydrates by amylase. It dissolves food, so nutrients can bind to those taste receptors, gustatory receptors, and that lysozyme already helps to cleanse the mouth with those antimicrobial properties. All right, so here's a salivary gland. I think this is actually really interesting when you actually look at the glands. This is a typical acinar gland. Okay, so if you remember way back with the epithelial cells, we talked about the different types of epithelial cells. This was the second one we talked about, a typical acinar gland. It's divided into two parts. The bulb part is called the acinus, and then the duct portion. So, just to be very clear, because this might be on your exam, primary secretions are very isosmotic. They're very plasma-like. So these primary secretions are very isosmotic, very plasma-like. So here's what a typical capillary looks like. This is supposed to symbolize a capillary. It's filtering, so the plasma is being forced out into the acinus. These are the primary secretions, and then they're modified in the duct portion. Okay? Usually what you see is sodium chloride reabsorption, so salts are pulled out, and the salivary secretions are usually hypoosmotic compared to the blood, compared to plasma. All right, so you also have these myoepithelial cells. They contract, which can regulate the flow through this area as well. And then finally, this is actually interesting. This is one of the only structures that are stimulated by both the parasympathetic and the sympathetic nervous system. By both. Okay, kind of interesting. All right. So I'm not going to go into too much about taste receptors. I know our lab actually did a lot of this when we were talking about the senses in week 3. Taste receptors include salt, sour, which is detecting pH, salty, sour, bitter, sweet, and umami. One of my favorites. Umami is a taste receptor that detects amino acids, proteins. So when you eat something savory, that's going to really activate these umami receptors. Okay? I think this is interesting as well. Taste receptors have actually been found in the small intestines. Can you believe that? In the small intestines. So whenever you eat something sweet, in the gut, it's detected by sweet taste receptors as well, and it inserts more sodium glucose cotransporters so that you can absorb more carbohydrates, more sugar in response to that sweetness. Which is why, think about that. I think I've already posted the PNAS paper that talks about this, which is why maybe you can't lose weight with Diet Coke and artificial sweeteners. All right, think about that. If your gut is actually detecting those sweet artificial sugar, it's going to insert more sodium glucose cotransporters so that you absorb more glucose in the face of this Diet Coke. Okay, so whatever you ate with your Diet Coke, you're going to actually get an elevated blood glucose sugar levels. Okay, something to think about. There's a PNAS paper that's actually posted. Take a look at that. So taste receptors, not only in the mouth, but also in the gut. All right, so swallowing. Here's the swallowing reflex. It's all coordinated by the medulla oblongata in the brain stem. It stimulates the appropriate sequence of contraction and relaxation, in particular skeletal muscles, sphincters, and smooth muscle groups. Okay, so you actually have a bolus of food here. You have voluntary phase, and you have an involuntary phase. Okay, so let's take a look at this in more detail. The two parts of the voluntary phase are this. Swallowing begins as the tongue actually pushes that food back to the back of your throat, called the pharynx. You're doing that. You're actually swallowing. You begin by pushing the food back with your tongue. And before swallowing, muscles keep the esophagus closed. The involuntary phase is this. That soft palate should rise up. Like I said, if you're drinking milk and you relax, then this whole system breaks down and milk comes out of your nose. But what should happen is when you eat that food, tongue pushes it back to the pharynx. The soft palate rises up to close off the nasal cavity, and the epiglottis bends down to protect the trachea, and then the food slides down the back into the esophagus. Pretty good? All right, how are we doing? We're doing really good on time. Okay, so this is the esophagus. Notice these different sphincters. This is really important here. You actually have an upper esophageal sphincter. UES is the acronym. You'll see this. Upper esophageal sphincter and the lower esophageal sphincter. So some people that have taken labs as well, even the animal diversity lab and CBS, you might know this as the cardiac sphincter. The LES, the lower esophageal sphincter, is also known as the cardiac sphincter. But in this class, I want you to know it as the LES, or the lower esophageal sphincter. Okay? Again, the esophagus, it has a lot of smooth muscle, skeletal muscle surrounding it. It really is just a tube to transport that food from A to B. But there is a lot of the coordinated sequence of contraction and relaxation in the upper esophageal sphincter, the esophagus, and the lower esophageal sphincter is necessary to deliver that swallowed food to the stomach. All right, so here's how it works. All right, so this actually gives you a really, really good idea of how the esophagus works. It's a little more complicated than just a tube. But the main thing to remember is the upper part of the esophagus is made of skeletal muscle, and the lower part of the esophagus has a lot of smooth muscle surrounding it. Now, think about that for just a second. Do you remember we talked about myasthenia gravis back when we were talking about muscle? Myasthenia gravis was an autoimmune disease that attacked nicotinic acetylcholine receptors, which basically rendered the patients, they had a lot of muscle weakness associated with it. So like I said, dogs and cats and animals, they can't tell you that they have generalized muscle weakness. They may just look super lazy, right? One way to diagnose dogs of myasthenia gravis is usually the skeletal muscle of the upper esophagus tends to relax abnormally. It's called megaesophagus. So a lot of times food actually starts to accumulate in this area. And when you look at it with an MRI, you can see that it's kind of really bulging out, right? Again, it's called megaesophagus. If a veterinarian actually sees that on the x-ray, they may say, oh, this is a really good indication that this dog has myasthenia gravis. That also tells you that the upper esophagus is under skeletal muscle control and the lower esophagus is under smooth muscle control. Now, I always think, is this voluntary, right? Can you actually, I don't think so. Can you manipulate this skeletal muscle? Do sword swallowers, do they have some trick that they can actually control the skeletal muscle? I don't know. I don't know what the secret is. All right, but just remember the upper esophagus is under skeletal muscle control, the lower esophagus under smooth muscle control. So let's take a look at this series of graphs because this actually tells a lot about the upper esophageal sphincter and the lower esophageal sphincter. All right, so on the x-axis, we're talking about time in seconds. And on the y-axis, this is actually measuring pressure. Measuring pressure. All right, these panels correspond to each of these locations along the esophagus. So when a bolus of food, a big wad, a bolus is just a big wad of food. When a big wad of food actually enters into this area right here, you can see it causes an increase in pressure, and then it returns as the food moves into the upper esophagus and the skeletal muscle controlled part. You can see it basically returns back to its normal pressure, 0. But the upper esophageal sphincter always has a basal tone associated with it. So it already begins at 30 millimeters of mercury. You can see before that bolus of food enters into the area, when it's right above here, there's a relaxation of the upper esophageal sphincter. And then the bolus of food enters, so you get a rise in pressure. And then it returns back to 30 millimeters of mercury. So there's a relaxation of the upper esophageal sphincter, and then the bolus of food moves through. So again, just remember that upper esophageal sphincter actually has smooth muscle surrounding it, and there is a basal tone associated with it. All right, so then the bolus of food moves through this part, the upper part of the esophagus, the lower part of the esophagus, and then that part that's under smooth muscle control. But take a look at the lower esophageal sphincter. Again, it also has a basal tone associated with it, but it relaxed way before that bolus of food entered into the area. Okay, so that's really interesting. This is an anticipatory relaxation. An anticipatory relaxation. So it relaxed when the bolus of food was way up in the upper part of the esophagus. And then the bolus of food moved through, and you can see the elevation in pressure and then back down to its original basal tone of 30 millimeters of mercury. Pretty cool, actually, that that lower esophageal sphincter relaxes before the bolus of food even gets into that area. All right, so a couple of pathophysiological states, both achalasia, this is called achalasia, and esophagitis are conditions where the lower esophageal sphincter fails to function normally. Okay, so I'm going to start with esophagitis first. Esophagitis, anytime you see itis at the end of a word, that just means inflammation. So this is inflammation of the esophagus. Esophagitis is a condition where the LES fails to maintain its tone. So a lot of acid bubbles up from the stomach and starts to cause inflammation of the esophagus in that region. Anybody know what this is? Yeah, it's GERD. Reflux. Yeah, it's GERD. Gastroesophageal reflux disease. Okay, it is a reflux disease, and it again causes inflammation. Sometimes, if it gets bad enough, it can cause bleeding and destruction of the esophagus. If it gets really bad, it can cause cancer, esophageal cancer. So someone that has chronic heartburn should really go into the doctor, get it taken care of. You don't want your esophagus to be destroyed like that, okay? So what happens if you have chronic GERD, right? Gastroesophageal reflux disease. What can happen is achalasia can happen afterwards. Okay, so what happens is scar tissue starts to build up. And then, if it gets really bad, you get to the point where the LES, the lower esophageal sphincter, fails to relax. Okay, so my uncle actually had GERD so badly that it turned into achalasia. And he had to go into the emergency room and get tubed, basically. Push that food into, it's very painful, it's very uncomfortable. And that's the remedy if it gets too bad. There are some drugs that help to relax it, but achalasia is really kind of a very uncomfortable disorder. So that's the difference between the two. I do have an interesting story again. There are two types, well, actually there is really one major type in the esophagus. It's called, this is the motility in the esophagus. It's actually called peristalsis. Anybody heard of peristalsis? Peristalsis. Peristalsis. Peristalsis is a coordination between circular smooth muscle and longitudinal muscle. And that's actually what happens in the esophagus. So what happens is there is a constriction of the smooth muscle by circular smooth muscle behind the bolus of food. And then the longitudinal muscle shortens to push the food down the esophagus. I always liken it to sweatpants. Okay, so bear with me. I actually thought about this years ago, and I thought this is exactly what happens when you has anyone lost the string in their sweatpants? Okay, when you try to restring it and it has that knot, what you do is you pinch the back of the knot, and then you take the fabric in the sweatpants and you push it along, right? That's kind of how peristalsis is. You pinch the back of that knot, and then you move the fabric of the sweatpants. Sometimes I think, does everyone understand that? Okay, perfect. All right. So let me just show you this actually. When my daughter was in first grade, this is years and years ago, I walked by her room like 10 o'clock, and she was crying. She didn't even come get me. She was just crying in her bed. And I said, "What's the matter?" She said, "I have a stomach ache." And I said, "Oh, what did you eat?" I was trying to figure out, did she eat candy before bed? Like, what was happening? And I said, "What did you eat?" She said, "I ate money." And I said, "You ate money?" And I said, "Was it a penny? Was it a dime?" And she said, "No, I ate the big one." And I was like, oh no. Okay, so we went to the doctor. They gave me this x-ray because I was like so impressed. So, see, you can see her spine, and you can literally take a quarter out of your back pocket, and it fits perfectly right there. Okay, so what happened was I took her to the emergency room, and she was having chest pains, right? She'd clutch her chest, and then she'd fall asleep. And she'd clutch her chest and fall asleep. And I was like, that is secondary peristalsis, all right? So she had it in her esophagus. It was stuck in her esophagus. And peristalsis was happening, right? Circular smooth muscle was contracting behind the quarter, and it was trying to push the quarter down, but it wasn't able to do it. So I told the doctor, that is secondary peristalsis. He said, perfect. And I go, I really want that x-ray so I can talk about it in physiology. So what happened? How did she recover from this? What she had to do, she was tiny too. They had put her in a little baby straitjacket, and they tied her arms behind her, and they actually had to tube her, right? And they pushed the quarter. They said once it got in her stomach, it would pass normally. I did actually have to try to dig it out and find it a day later or so, which I did, because that stuff doesn't really bother me. Okay, but that's actually how that happened. So anyway, very good example of peristalsis in the esophagus, okay? So, good stuff. Let's see. That is a good place to end, actually. We'll actually start with the stomach and digestion on Wednesday. All right.